Hello everyone, I am Gizacho Teshome from Ethiopia, PhD student at Addis Ababa University. I would like to present my research paper which has been published last year. It's entitled on influence of irrigation infrastructure and water quality on fish assemblage in Lake Tana territories, Northwest Ethiopia. Authors from University of Gondar, Addis Ababa University, University of Newcastle, Australia, and Barda University. Here listed authors. The presentation has an outline, introduction, objective, methodology, result, indication, and conclusion. When I come to the introduction part, lakes hold nearly 90% of the world freshwater resource, which represent a key ecosystem for aquatic biodiversity. Lake Tana is one of the largest lake in Ethiopia. It is a highland lake. It is located in northern western Ethiopia. It has been recognized as a high conservation value regionally and globally. Based on this, UNESCO has listed it as a biosphere reserve in 2014. Here, the map show from the African map, the country Ethiopia located in the horn of the Africa, the northern western part of the country, the lake is located. The lake is recognized as a habitat of a lot of fish species, more than 20 fish species inhabit Lake Tana. Among these species, 21 are endemic to Ethiopia. Unfortunately, due to have a lot of anthropogenic factors, agricultural and urbanizations and other potential driving forces, currently a number of species has been registered, you see in a red list, with a range from data deficient to endangered. Some of the species, about 4 Labeobarbus species are registered as vulnerable. So the lake needs need urgent management, particularly to prevent endemic Labeobarbus species in the country. Lake Tana is fed by more than 40 small seasonal and 7 large perennial tributary rivers that only have one river out from the lake, which is known as Blue Nile River. The head of the Blue Nile River is Lake Tana. It is here the picture shows a natural waterfall which is found around the lake. Lake Tana Basin under severe stress from agricultural and the population growth have a lot of driving factors which affect the lake ecosystem and the fishery biodiversity. For instance, expansion of irrigation dams along Lake Tana territories, excessive stream sedimentation and riparian erosion, as well as sand mining in the river channel are some of anthropogenic factors which affect the migratory fish assemblage structure, as well as spawning ground is important variables which support the fish spawning grounds. So based on this background information, the study was examined the following main objectives. The first one is how migratory fish assembly structure varied over the time in between tributaries. And another objective was how irrigation infrastructures affects the abundance and the distribution of adult and juvenile fish in the tributary rivers. And the last, determine which physical chemical water qualities affect the adult fish and juvenile fish distribution abundance in the rivers. Here is a study material and meter section. When here is a map of Africa and here is Ethiopia, Lake Tana, northwestern part of the country. Here are the 5 tributary rivers were selected purposefully based on their suitabilities for fishing, and as well as more spawning grounds available sites. Here is a study sites where Shini rivers with irrigation canals, and there's a dam. Here is a Guanta River. Here are juvenile fish sampling and field observations. Look here how the fish samples were collected. Those sampling sites, 5 rivers were selected purposefully based on different anthropogenic factors and natural factors. Then those sampling sites were stratified based on the sand mining and non-sand mining sites. Here is non-sand mining site. Here is sand mining sites. How the river vegetations, large vegetations were deforestations and also river shoreline erosions and river bed destructions due to the presence of high intensive irrigation sand mining activities. These are the way how the sampling sites were stratified. Field sampling sites were selected based on the channel access and the presence of sufficient sampling area, as well as availability of expected spawning sites characters such as sands and gravel beds. Based on these characters, those sites were selected purposefully. There are two types of fishing gears were used for fishing activity: multi-filament and monofilament gill net. Beside to fishing, some physicochemical parameters were measured in situ using portable multimeter. Then after collect the data were analyzed using different statistical analysis to show the assemblage structure of migratory fish species into those selected sampling sites and the water quality impact and the irrigations. Primer 7 and the space software were employed for analyze the data. When I come to the result in discussion section, adult fish abundance, diversity, and simply structure were analyzed. A total of more than 100, 300 fish specimens under these 13 species were sampled over the 5 tributaries and 5 sampling trip. Dominantly by Labeobarbus species, 9 species, 95% of the total catch were genus Labeobarbus. Almost 9 species were recorded. Among 9 species, Labeobarbus intermediates and Labeobarbus principals were the most abundant species, 72% of overall the specimen. The level of dissimilarity between tributary pairs was highest for the Shini and common, 55.6, and the Javarna and common also the followed 52%. When we see non-metric dimensional scaling of adult abundance showed variation in assembly structure both within and between territories. In contrast, no significant difference in adult assembly structure were detected between sampling trips. Analysis of similarity detects significant difference among territories at 0.01 significance level. Here, the data shows between sampling sites here. When you see the irrigation infrastructure impact on upstream and downstream migration, we found the abundance in the diversities show significant difference above the dam and below the dam. Why? Due to the presence of this dam, which affects or barrier for upstream migrations. Here show the black color in the picture showed those species found above the dam, and the greens shows the gray color show those species is abundance in the diversity below the dam. This is obviously different researchers also agree that some irrigation dams and weirs affect upstream and downstream fish movement. When I come to the water quality from those rivers, sampling sites showed significant difference in different physical-chemical properties, which might be due to have different catchment and different activities there in different environment or in different rivers. Water physical-chemical characteristics appear to explain some of the variability in adult assemblage structure across the study territories and sampling interval. In combination with total dissolved solid, pH, secretives, it explained almost 44% of the variation in assemblage structure. Here, the picture shows how the algal blooms created above the irrigation dams, which affects the fish distribution and abundance. Conductivity and turbidity related variables like secretives, total dissolved solid had the strongest association with both adult assembly structure and individual species abundance. In contrast to this study, some studies showed a negative association with the water quality with the fish abundance and diversity. Here, studies elsewhere have linked increase in stream turbidity and suspecting materials level to various anthropogenic sediment in disturbance. This also confirmed by other studies. The last we have discussed the data and those observed different potential anthropogenic factors in the study sites. For instance, agricultural practice and vegetation clearing have resulted in substantial erosion problem across the Tana region. High level of downstream siltation was reported in the catchment of Rip and Gumara. Fluvial sand and gravel extraction also occur along most Tana territories. A lot of important spawning ground routes and important support materials were damaged, which maybe affect those migratory fish species sustainability. Sedimentation and turbidity during sand mining have consequences in fish of the Tana territories. For unfortunately, during the peak time of spawning period, there was high pressures of sand mining in each tributary rivers, which maybe affect important materials, important substrates in the riverbed, in the water quality, which affect the migratory fish habitats. Loss of spawning habitats, lower egg retention and survival in smothered gravel beds, as well as reduced visual activity, acuity growth and survival in larvae. Finally, we found Lake Tana territories to compromise a suite of unique fish biodiversity units. It inhibit a lot of endemic migratory species there. So irrigation dams disrupt upstream spawning migration, Lake Tana fish, and the downstream movement of juvenile also substantially impacted. Direct assessment how poor water quality may be inflated through catchment erosion are urgently needed to develop effective intervention strategies. Establishing greater understanding how irrigation infrastructure and water quality impact the early life history stage of fish within tributaries, as well as effective conservation measure at the lake scale to all endemic species, highly recommended. Thank you very much. Those helped me during conduct the research and publish this paper. Professor Glenn Wilson and Professor Abebe Getao, Rafford Foundation in Bahar University for the fund support. And then finally, I'd like to the American Fisheries for giving this chance. Thank you very much.